Hi, I'm Morgan Edwards, and I'm a freshman who currently attends North Carolina Agricultural and Technical State University. I'm majoring in biology, and I'm here to talk to you about my protein structure project. My specific protein is the melanocortin 1 receptor. The melanocortin 1 receptor can also be known as the MC1R, and it's located in the plasma membrane of a specialized cell called the melanocyte. The melanocyte is usually found in different organisms such as mammals, and it's a polytopic helical protein with 7 transmembrane helices, as you can see right here. Inside the protein, you'll find different codons, which are represented by the beads, and it'll make up different amino acids. It has an extracellular portion and an intracellular portion. That can support the fact that it's represented to be a receptor. Because when you look at what a receptor is a protein that can be inside or on the surface of a cell, and when a substance attaches to it, it'll be stimulated and it'll cause an effect. Analyzing how the melanocortin 1 receptor functions, a melanocyte stimulating hormone, which can be generalized as a ligand, will attach to the extracellular portion of the MC1R, and it'll be stimulated and it'll produce CAMP. CAMP is specifically a messenger used for intracellular signaling, and it'll produce 4 different genes. When the 4 genes are expressed, then eumelanin is produced. Eumelanin is a dark pigment that's produced through melanogenesis by the melanocyte cell, and the MC1R impacts the cell as a whole by starting up the process to create eumelanin. The production of eumelanin tends to make a person's or an animal's hair a darker shade. The MC1R is part of the integumentary system, and that's a system responsible for being a physical barrier for the body, and it's associated with things as your hair and your nails. Now, to be really specific, I would like to draw attention to the mutation that occurs in the melanocortin 1 receptor itself inside of a beach mouse. Originally, majority of beach mice had a darker shade fur. As time went on, coastal mice, they became a lighter yellowish tan color. The reason that happened is because of a mutation that occurs inside of the melanocortin 1 receptor. The mutation, it occurs in the first loop of the intracellular part of the MC1R, and it's amino acid number 67, and it's argine. And argine is, out of the 19 amino acids, it's pretty large. The side chains of arginine is methyl groups and amine groups, and that makes the side chain of the arginine polar, meaning that it's hydrophilic and it interacts with water. It's also positively charged, so it'll be able to interact with aqueous environments and form hydrogen bonds and fold a certain way to interact with like substances. Now, in order for the mutation to occur, arginine is swapped out and replaced with cysteine. Cysteine is an amino acid, and it has different properties than arginine. It's a relatively small amino acid, and the side chains are made up of a sulfhydryl group and a methyl group, which makes it polar, but it's neutral. So both amino acids are polar, but their charge will determine how they interact with different substances. Because of the mutation, when the MC1R is stimulated by the ligand that will attach to the extracellular portion of the protein, it will no longer correctly function. And now it won't stimulate and produce enough CAMP, and the CAMP genes won't be expressed to create eumelanin. Instead, it causes the cell to create a color called pheomelanin, which is like a yellowish tan color. Now, this really explains why the coastal mice are no longer having dark brown fur and instead having a tan lighter fur. The MC1R becomes less active because there's a small amount of CAMP being produced instead of the eumelanin being produced. Now pheomelanin is produced, and it turns everything a lighter color. Some limitations that occurred in my project is you can't necessarily see the important portions inside of the protein that occur, like the production of CAMP, which is very important when looking at the production of eumelanin. And another limitation is it's clearly bigger than the actual protein on the cell, so it's blown out. And my last limitation is that it's not necessarily this organized. Instead, it'll be folded different ways along along the membrane, because of the different side chains that are made up from the amino acids, and they'll interact with like substances, and it will repel from unlike substances. I really hope you enjoy my protein project.